Thank you, Gil. So, developing drugs is very difficult. The pharmaceutical industry spends an enormous amount of money every year on trying to develop drugs. Only very few actually make it all the way. The cost per drug is very high. And so there's been, over the last 10 years, a real acceptance of the idea that if you want to have a higher probability of taking a drug all the way, if there's strong genetic evidence underlying your drug, then it's going to have a higher chance of success. And so really, you do need to pursue, if you want, you can't pursue modern drug discovery without incorporating human genetics. And as Gil said, Regeneron have really been embracing this idea. So over the last 5 years, they have built what I think is a really state-of-the-art sequencing lab, as fully automated sequencing and genotyping, sample preparation workflows, and now can scale to over 500,000 exomes a year with an analysis framework in the cloud. And so to date, we have sequenced over 600,000 exomes and run nearly approaching 700,000 samples on genotyping arrays. We have upwards of 70 collaborative projects with academic groups and healthcare systems. And we looked the other day how many data sets, GWAS, XWAS, we actually have, and we have over 100,000 in-house now, combining over cohorts, ancestry, different technologies and analysis methods. And this is a tremendous resource for us to develop new targets, to help the development of existing drug programs and reposition existing drugs, finding new indications for them. But as I said, we've got this lab that can exome sequence at scale. The exome represents 1 to 2% of the genome. Every gene has a mix of exons and introns, and it's those exons that get transcribed and translated into proteins and have a major impact on biological pathways. So what we're really interested in is mutations effectively that disrupt these very key parts of the genome. How do we go about finding those mutations in the most efficient, accurate way possible? Now, you can use genotyping chips, but the problem with genotyping chips for exons is you don't tend to get that many of them. Genotyping sites in exons, they're mostly common variants generally, and as various people have said, for the rare variants, they can be very hard to genotype. There's imputation, which is a computational method that can make predictions at many more sites. In Biobank, we increased the number of sites by 100-fold or more, and that works very well mostly, but it is dependent on the reference panel size and quality. These reference panels are always increasing in quality, so this will always, I think, be a part of this procedure. But really, if you want to go in detail into the exons, you need to actually use sequencing. And this gets you even more sites and very accurate calls at those rare sites. But we don't just want all of those sites. We're really interested in the ones that disrupt function, the so-called loss of function variants. And so you can make predictions of the function of those variants and use that in your analysis. You could go even further. I think Kari will talk about that after me, for whole genome sequencing, but sequencing is expensive. If you're going to pick 1 or 2% of the genome to sequence, I think it makes sense to start with the exome first. And so at the end of 2007, UK Biobank, Regeneron, and GlaxoSmithKline came together to start a project to sequence the full UK Biobank, exome sequence the full UK Biobank. And so in June last year, the first 50 exomes were completed, and that data was released to approved researchers in March this year. And the timeline moving on from that, so moving on, the rest of the sequencing is being done by a consortium of 8 different pharmaceutical companies, and the timeline for the completion of the sequence is shown here. So the full 500,000 we predict now will be done in the first quarter of next year. That data, to start with, will be available to these partners and will be released by UK Biobank 12 months after these dates. So what have we found so far? So I'm going to talk a little bit about the 50K data set that's been released. We did find a very large number, around 4.7 million coding variants. The vast majority are very rare. There's a very large number of missense mutations and loss of function mutations. One interesting question is to ask how many genes have a loss of function mutation in at least one person. And so you can see that this data point on this graph here, we find, given this very specific definition of what a loss of function mutation is, that almost every gene, 97%, we can find at least one person who has a loss of function variant in those 50,000 samples. If you then ask the question, how many genes are there where you can find 10 people, at least 10 people with a loss of function variant, then it's about 70%. And so we can actually then subsample, resample, smaller sample sizes to generate these curves. So if we had 1,000 people, we might get about 7,000 genes where we can find one loss of function variant. What we're interested in making predictions moving forward. When we get to 500,000, how many genes will have a loss of function mutation? Just quickly, we did compare this number to an existing database, the NOMAD database. We applied very similar processing to that database and it's a similar size, and we got a similar number, so we're reasonably confident in our analysis. It does depend on these details here. So how can we make predictions? Well, if you found 13,000 genes that have at least 10 people with a loss of function variant in 50,000, well, it makes sense that if you increase the sample size by 10, you'd find that many genes, but with greater than 100 people with a loss of function variants. And that essentially means we can take this data point here and predict it forward for what that blue line would be. And so you can make lots of predictions from these curves and then extrapolate these numbers. And so these are telling us then that in the full sample, we expect 13,000 people roughly to have at least 100 people with a loss of function variant. But there's a caveat to that. We think probably these are overestimates. And the reason we think that is if we try to interpolate within our data set, we get slight underestimates. And so it gets even worse if we use this black line here. And so the interpolation suggests that what we've done is slightly overestimating, but is still, we think, reasonably accurate. So comparing to the imputed data, we do find a lot more variants. Only 13% of the exome variants that we've uncovered exist in the UKB imputed data. We have a 12-fold increase in the number of loss of function variants. So for the rare ones, it's 234,000 versus 18,000, with nearly 12,000 shared. And for the missense variants, it's a 6-fold increase. One interesting thing that we've been looking at is focusing in on a group of genes identified by the American College of Medical Genetics. So these are genetic conditions and associated genes where there's strong evidence that you can make you can mitigate that condition if you find a mutation in one of those genes. And so we looked at those genes in our 50K samples. We found variants in 48 of those genes. Some of them, the BRCA genes, the LDLR for familial hypercholesterolemia, PMS2 for Lynch syndrome, KCNQ1 for cardiac arrhythmias. And overall, we estimate that about 2% of the full UK Biobank population carry a loss of function variant that is said to be medically actionable. And of course, we've been trying to use this data set to uncover new targets. The first thing we did was look at positive controls, genes that are well established in the field, that have a rare loss of function burden. So there's some binary phenotypes here with associated genes, some quantitative phenotypes here with associated genes. And in the 50K data set, we see very strong signals of association, which then also because we have the 150K exome data in-house already, we can look and to see if those signals stack up there as well, and they do. So this is encouraging that this strategy can find these rare loss of function signals. And then, of course, we've been looking to find novel variants. And so these are results from the 50K data. And so we found several binary phenotypes and quantitative phenotypes. So this one here is for varicose veins. The PIZO1 gene has a very significant association, which stacks up when we also look in the 150K. Okay, so this paper is available if you'd like to learn more about it. And I'd just like to thank everybody from the Biobank, the participants, organizers, and researchers, everyone involved from Regeneron and GlaxoSmithKline for making the 50K phase 1 project and paper a success, and the ongoing work by all of the consortium members to make this a reality.